I'm Dr. Plant.
I'm Dr. Luna.
And I am Dr. Troy.
And
this is cell signaling. Cell signaling is crucial in cells because it is a necessity for the cell to change what it is doing based on the conditions it can't control. To do so, the cell must go through three stages: reception, transduction, and response. Whether it's long-range, hormones, or short-range, local regulators, the cell has a systematic way of sending the signal and getting it across.
Within a cell, there are three types of signaling. Under local regulators, the cell can either conduct paracrine signaling or synaptic signaling. In paracrine signaling, a secreting cell acts upon the target cell by releasing a local regulator into their fluid. Because of this, they begin to change and react. In synaptic signaling, a nerve cell releases neurotransmitter molecules into a synapsis, which is a narrow space between the transmitter and receiver. A hormonal method of signaling is cells secreting hormones into body fluids that have the ability to reach all the cells because of blood flow. This method can only work because of the correct reception.
What is reception, you might ask? Reception is the target cell's detection or chemical signal that binds to a chemical protein. These chemical signals that bind are known as ligands. One kind of receptor is a G protein-linked receptor. When a signal molecule bonds to the G protein, the GDP molecule is instantaneously displaced by GTP. The active G protein binds and activates the needed enzyme, which can then transfer a response. The G protein is then goes through hydrolysis of its GTP and dissociates from the enzyme, becoming available in case it is needed again by the cell. Another way that cell reception can be occurred is through the protein tyrosine kinase. It exists as a single polypeptide in the plasma membrane. When a signal molecule attaches to two binding sites of the molecule, they instantly aggregate, forming a dimer. When this happens, phosphates are added to each tyrosine receptor. The phosphorylated dimer is now both an enzyme and a substrate. Now intercellular proteins can bind to the tyrosines to become activated, initiating the signal transduction phase. A third and final method to receive a signal is through ligand-gated ion channel receptors. These channels are protein pores that open or close, allowing or blocking the flow of ions. When these ions change in concentration within the cell and force the cell to These ions change in concentration within the cell and force the cell to trigger a cellular response. Check.
Protein.
Protein.
Go.
The transduction phase is important because once the signal is received, it must be converted into a signal that represents the correct cellular response. This process is also known as the signal transduction phase. A ligand binds to a receptor and triggers a chain reaction that does not physically pass along the molecular message, but converts it to another form. This can be seen in the phosphorylation cascade, where a series of protein kinases successfully add phosphate groups to the next thing in line, activating it. The enzymes soon remove the phosphate, keeping the path open for a new signal. Secondary messengers are used as smaller parts of the signal transduction phase and involve a small non-protein molecule. One second messenger is CAMP. CAMP binds to the ligand that binds to a receptor. Conformation changes, making the G protein activated. Now the enzyme activated, adeno, can convert ATP to CAMP that binds and activates the cytoplasmic protein that is needed. Kinases then can propagate the message by phosphorylating various other proteins for other proteins, leading to cellular response. Hormones can use this form of transduction. Calcium ions and IP3 are also secondary messengers that allow for specific responses based on concentration. The process is initiating by the binding of a signal molecule to the gated protein channel or tyrosine kinase receptors. The enzyme is activated and cleaves to a phospholipid called PIP2 into DAD and IP3. IP3 quickly diffuses through the cytosol to bind into a gated calcium channel in the ER, causing calcium ions to rush in, activating proteins by signaling other pathways. In response to a signal, a cell may regulate activities in the cytoplasm, or transcription in the nucleus. Signaling pathways regulate enzyme activity and cytoskeleton arrangement. Other pathways regulate genes by activating transcription factors or proteins that turn on specific genes. Cells can also amplify and specify the cell's responses to signals. A catalytic protein amplifies the signal by activating multiple copies of the components of the pathways. Each combination of proteins amplifies it up to 1,000,000-fold. Pathways help branch out and help the cell to coordinate signals.
I'm Plant.
I'm Zarumba.
And I'm Luna. And we hope you learned a lot about cell signaling from this video.